Hello students, I am Jyoti, working as lecturer in microbiology at Government College for Women, Srikakulam. Today I am going to present a topic on industrial production of microbial therapeutic enzymes. This topic is dealing with BSc final year microbiology students. First of all, we would like to know what are enzymes. Generally, enzymes are the biocatalysts that catalyze the metabolic and biochemical reactions in the living organisms. If we want to know what is the chemical nature of enzyme, enzymes are generally macromolecules made up of proteins. Now, enzymes are proteins, and there are some enzymes are also which act as a where RNA act as a biomolecule. Here these type of enzymes are referred as ribozymes. Not only RNA act as enzymes, even some antibody also serves as enzymes, and these enzymes are referred as enzymes. Enzymes are required for our day-to-day process in metabolic reactions, in digestive reactions. Now, functions of enzyme is basically the function of enzyme is to convert a substrate into a product. Here, for example, if we get to know amylase is an enzyme. Here, amylase is responsible for conversion of a substrate into a glucose as a product. Similarly, in our day-to-day type, we take different types of diet. In diet, it includes proteins, carbohydrates, lipids, and these are all macromolecules. These macromolecules should be broken down into monosaccharides. For this thing, the enzymes are being used not only for digestive process, even for the metabolic process, like in glycolysis process, TCA cycle process, enzymes are generally used. So, if we get to know about the functions of enzymes or the properties of enzymes, enzymes are highly substrate specific. Substrate specific means enzyme like amylase enzyme, it acts only on starch as a substrate. It uses starch only as a substrate, but not proteins as a substrate. So, every enzyme is having a particular substrate. So, that enzyme should act on only that substrate, but it shouldn't act on other substrate. And the enzyme activity, what do you mean by enzyme activity? Means it converts a substrate into a product. When it takes over the biochemical reaction, it is influenced by the various factors like pH, temperature, and substrate concentration. pH means every enzyme, since enzymes are produced within the living organisms like plant, animal, and some microorganisms, they are maintained at their optimum levels. The optimum pH should be maintained. Generally, enzymes may require acidic pH, basic pH, or neutral pH. So, whatever the enzyme we are working with, or whatever enzyme is acting in certain conditions, it is influenced by certain pH, and it will work effectively at that pH. And that pH may range from 1 to 5, or it may be act as a neutral pH, or it may be act at the basic level pH, that is from 8 to 14. And other second point is enzymes are influenced by the even temperature parameter. Every enzyme is influenced by the temperature parameter. Temperature parameter means generally enzymes are active at 25 to 30 degrees temperature. Certain enzymes produced within the human beings, enzymes secreted within the stomach, pancreas, are active at the 37 degrees centigrade temperature. So, we can say that enzymes are best at its optimum temperature. So, however, certain times when enzymes go beyond their optimum temperature, it may lead to the denaturation of the enzymes. Here I am saying the denaturation topic. Why? Means enzymes are proteins. Since proteins are also subjected to denaturation at certain temperature levels, even enzymes undergo denaturation. Again, the third factor is enzymes. The activity of enzymes activity is influenced by the substrate concentration. Substrate concentration means if there are 10 units of enzymes, it should be influenced by the 10 units of substrate. Minimal quantity of enzyme or maximum quantity of substrate does not carry out the reaction. So, enzyme activity is also influenced by the substrate concentration. Now, what are the sources of therapeutic enzymes? Sources of the therapeutic enzymes. Therapeutic enzymes can be extracted from the plants, it can be extracted from the animals. Animals means in the humans, they are synthesized within the digestive, in the stomach, in the pancreas, in the salivary glands. Commercially, for commercial production of therapeutic enzymes, microorganisms are employed. Microorganisms such as bacteria, yeast, actinomycetes, fungi are employed for commercial production of therapeutic enzymes. Therapeutic enzymes. Why microorganisms are employed as for commercial production? Because we can say that microorganisms are very readily available. They are available in our natural microflora, in our soil, in our water samples, and in many sources microorganisms are easily available. So they are referred as a cheap source. Even since they are readily available, they can be reusable, and they are economically feasible. And compared to the other sources, microorganisms tend to give higher yields, and it is easy for optimization of their growth. So nowadays many researchers, many universities are concentrating on the exploiting microorganism for the source of enzymes, and it has become a billion-dollar business in the world also. Not only therapeutic enzymes, even other enzymes are exploited from the microorganism. We can say that there are uncountable microorganisms in our soil microflora. So students, if they take this research for exploiting or for isolating the microorganisms which produce enzymes, it can be deal with a good research points. Now, when I am saying about therapeutic enzymes, generally therapeutic enzyme means enzymes that are dealing with in the treatment of any particular disease. Generally nowadays for any treatment we go for different treatment methods like drug therapy, radiation therapy, and etc. Now since enzymes are biocompatible with the human, enzymes can be biocompatible with the human bodies. Even scientists are exploiting these microorganism enzymes as for the treatment purposes. Now what is the properties? How these therapeutic enzymes work means generally they have high efficiency. Here high efficiency means they have low Michael-Menten constant and high velocity max. When even at the lower substrate concentration, enzymes work very efficiently. Even when we isolate these enzymes from the organism, they are of high purity and of high specificity. Here high specificity means they work on the particular substrate only without inhibiting with any inhibitor. So to conclude this topic, I would say that enzymes in our day-to-day life or enzymes can be exploited. Enzymes are used for enzymes acts as a biocatalyst in all living systems. Enzymes can be used for therapeutic purpose. Initially till now enzymes were exploited only for production of antibiotics, for penicillins, like penicillins, for drugs, and for alcohols, for beverages and other kind of enzymes. Now these therapeutic enzymes has been become a wide global research point, and many enzymes are going to be exploited, and many enzymes going to be studied. Now we can go for the next slide, next topic of the unit. Next topic means what are the type of enzymes and how these enzymes are isolated from different microorganisms. We will be studying about these enzymes. Types of therapeutic enzymes. Types of therapeutic enzyme means since humans are infected or associated with different type of diseases, these enzymes we should have different type of enzyme for treating different type of diseases. People are suffering from coronary heart diseases, people are suffering from cystic fibrosis, people are getting infected with blood-borne pathogens. Although we have many drugs and many antibiotics, we are not able to cure the disease, and even it is become a cost-effective process. So based upon the discovery, based upon the isolation of many researchers, we have identified there are innumerable number of enzymes which are having their therapeutic applications. Now first enzyme we will study about the lipase enzyme. Lipase is a general enzyme. It is a extracellular thermostable enzyme that catalyzes the hydrolysis of ester from the glycerol. As the name suggests, lipase enzyme generally acts on the lipid. So till now lipase enzyme has been isolated only for industrial purposes only, for industrial production only. But now lipase enzyme can also be exploited for the therapeutic use. Now lipase enzyme, for isolation of lipase enzyme, many bacteria, fungi, yeast, actinomycetes are employed. Commercially they are isolated from the Candida, Pseudomonas, Bacillus species. Now we can say that lipase enzyme is used for the treatment of malignant tumor. Malignant tumor means a uncontrollable tumor. Generally nowadays tumor has been treated with certain antibiotics or radiation therapy. Antibiotics may give some certain kind of side effects, and radiation therapies may also give rise to certain kind of mutations. But if we employ the enzymes, this can be it may not lead to any side effect, and it is very cost effective process. Along with that, lipase enzyme has been also employed for the treatment of a coronary vasodilator. It acts as a coronary vasodilator, and it is also used for in the treatment of it acts as a digestive aid in gastric allergies. Gastric allergy means certain people are susceptible to the, means they cannot digest the hard foods, certain hard foods, because their stomach or their pancreas does not secrete those kind of enzyme. In certain cases this lipase enzyme is supplemented as a syrup and it is given to a patient. This lipase enzyme facilitate in the easy digestion of the lipids present in the food material. Now another group of enzyme is referred as a chitinase enzyme. Even chitinase enzyme can also be used as a therapeutic enzyme. Again I am telling, therapeutic enzyme is these enzymes are employed for the treatment of any kind of infections or diseases suspected in the human beings. Now what do you mean by chitin? Chitin is the most abundant polysaccharide after the starch. It is present in the cell wall of some pathogenic fungi and in the exoskeleton of insects. Now chitinase, what is the nature of chitinases enzyme? Means chitinases are the hydrolytic endoenzymes. Here endoenzymes means certain group of enzymes can be secreted by the microorganism as exocellular enzymes, and certain group of enzymes can be secreted as a endocellular enzymes. Chitinases enzymes are generally employed for breaking down the chitin component in the cell wall of a pathogenic organism. As we everybody know, cell wall is a basic component of any microorganism. Without cell wall, that microorganism structure has been lost. So if chitinase enzyme is used for the treatment in pathogenic in infection suspected with microorganism having chitin in their cell wall, this chitinase enzyme can be employed, since this chitinase enzyme tend to hydrolyze or break the cell wall of pathogenic microbes containing chitin as their component. Now the chitinase enzyme is commercially produced from species such as many fungal species such as Aspergillus, Trichoderma, Fusarium, Aeromonas, Serratia, Vibrio, Streptomyces and Bacillus. Chitinase has been largely used and largely exploited. Why? Because it is having a massive antimicrobial activity, especially it serves as a antifungal agent. Now we can also know that chitinase induces innate immune response against asthma and infections with pathogens coated with chitin. So by seeing these applications or by seeing these types of enzymes, it is interesting to know that microorganisms, apart from causing infections, microorganisms are also having the ability to cure the infections. So microorganisms having the dual property. In medical microbiology, generally we studies that microorganisms are responsible for certain kind of infections. Microorganisms are not good for human, but these same microorganisms are also providing us with the remedy how to cure the infection by their ability to produce different types of therapeutic enzymes. Now let's move on to next enzyme. Next enzyme is a collagenase enzyme. Collagenase enzyme. What do you mean by collagen? Collagen is the most abundant protein. protein known on the earth. This collagen protein is a structural protein present in the skin, bone, muscle and other connective tissues. So, why our bones are very strong means we are supported with the collagen as a protein. Now, collagenase are the enzymes that break the peptide bond in the collagen. Now, commercially collagenase is produced from species of Vibrio, species of Clostridium, Bacillus and Klebsiella. Why what is the role of collagenase enzyme means collagenase enzyme helps in the skin repair, removes the dead skin tissues, helps in the treating burns and skin ulcers. So, after that collagenase we will be dealing with the asperginase enzyme. Asperginase enzyme, as the name suggests, asperginase enzyme acts on the aspergine amino acid. Basically, asperginase enzyme is used for in treatment of in cancers or in the tumors. It acts as a anti-tumor agent. Commercially, asperginase enzyme is produced from species of Pseudomonas and species of Aeromonas, Erwinia, Xanthomonas and E. coli. Now, since asperginase is having anti-cancer property and it also used in treatment of acute lymphocytic leukemia. Leukemia is a kind of blood cancer. Now, if we want to know what is the mechanism or what is the mode of action of asperginase enzyme means asperginase enzyme acts by reducing the availability of aspargine amino acid. Aspargine amino acid is a basic amino acid required for the growth of cells. When this basic amino acid has been depleted, the tumor cells doesn't grow. So, asperginase enzyme can be used in treatment of tumors by reducing the aspargine amino acid. Now, till now we have studied that enzymes there are what are enzymes, what are types of enzymes, what are the sources of enzymes. Now, we already know that sources of enzymes means it can be plants, it can be animals, it can be microorganisms. Commercially, microorganisms are exploited. Now, we will study how enzymes are commercially produced or how enzymes are produced in industries. Production of therapeutic enzymes is our next subtopic. So, generally enzyme or any industrial products are produced by the fermentation process. Fermentation process takes place in the microorganism during the different phases of growth, starting from the lag phase to the stationary phase. When the microorganism comes under the stationary phase, due to depletion of nutrients, it will tend to release certain end metabolic products. From that, enzymes are one of the metabolic products. If we study in industrial microbiology, there are different types of fermentation methods employed in the production of therapeutic enzymes. Not only therapeutic enzymes, any kind of enzymes can be produced from these kinds of methods. So, first type, generally microorganisms are grown by process of batch fermentation. If we know about what do you mean by batch fermentation, in batch fermentation generally all the nutrients are fed into the fermenter and the process may take place for 7 days or 10 days. During this batch fermentation also even the end product should be removed. Apart from batch fermentation, in industrial microbiology or industrial production, we also have the continuous fermentation. In continuous fermentation, the medium is continuously added into the fermenters, so that the microbial growth is maintained continuously and the products are produced continuously. Apart from this continuous and batch fermentation, we also have a surface fermentation. In surface fermentation, the microorganism or the inoculum is grown at the surface of the substratum or the medium. And coming for the substrate fermentation, in substrate fermentation, production of enzymes takes place within the medium, in the liquid medium. Compared with the surface fermentation, submerged fermentation provides many advantages. Submerged fermentation gives higher yields, give higher purity, and submerged fermentation is free from the contamination. Apart from surface fermentation and substrate fermentation, we also have a solid state fermentation. What do you mean by solid state fermentation? In solid state fermentation, the substratum used is completely solid, means it should be free from the moisture. Generally wheat bran, molasses, sugarcane bagasse, fruit peels, vegetable peels can be used as a substrate. What are the advantages of using agriculture waste as substrate? Means they are easily available and they can be reusable. So in solid state fermentation, the inoculum is spread over the fermentation, and the solid substratum act as a medium for the organism. The process may takes place for 7 to 10 days depending upon the growth of the microorganism and its secretion of its enzyme. And the fourth method and commercially more viable method is recombinant DNA technology. Recombinant DNA technology provides many advantages compared with the other fermentation methods. Recombinant DNA technology provides advantage of strain improvement. Through site directed mutagenesis, we can improve the activity of a gene responsible for production of that particular enzyme. So in the what are the steps involved in recombinant DNA technology? Generally in recombinant DNA technology, we will tend to insert our interested gene into the vector, and that is transferred into a host that will give rise to a multiple copies of genes. Now enzymes, we have studied everything about the enzymes. If we again recap how enzymes are produced, means enzymes are secreted by the genes during the translation process. Now if we generalize the what are the steps in the enzyme production includes, means isolation or screening of the microorganism. Microorganism here means high yielding strain. High yielding strain means there are many microorganisms in soil microflora. There are many species, but which species of particular genera gives us higher yield? Higher yield in the sense means high units of enzyme, and that enzyme should be highly efficient. Highly efficient means it should have a high turnover number. Yet high turnover number means it should able to convert the substrate into product within the very less time. So we should screen for the high yielding strain that is giving us a highly efficient enzyme. Now what are the samples? Mean we can go for regular soil samples or water samples as our sample. Screening means from the group of microorganism we are selecting the best strain. Now preparation of inoculum. Preparation of inoculum means the organism isolated from the soil sample should be inoculated into the general nutrient agar media in the laboratory scale. After that, after the organism tend to grow, it should be transferred into slants and it should be maintained. After that, the medium designed for that particular enzyme growth should be made. Every enzyme differs in the nature of the media. That media preparation we can refer from the research papers and we can discuss, and if we want to perform the particular experiment in our lab for production of particular enzyme, we should follow the protocol of that production of particular enzyme. We should prepare the media, we should autoclave it, and we should pour those plates and we should inoculate our sample. As we know, after 24 hours we will be getting the colonies. Through that colonies, again it should be transferred into another fermentation media. Now preparation of the growth media. Second step is the preparation of growth media. Here I can call as a growth media or the fermentation media. Growth media or fermentation media means the organism should be grown in a particular growth media. The media should have all the growth parameters for the growth of the microorganism. Like vitamins, carbohydrates, nutrient supplements, salts, everything. Even the optimization of growth medium should be maintained, like pH, temperature, water content, its aeration capacity. Generally in automated fermenters, all the temperature, pH, oxygen parameters are maintained in industries. So when we perform at the lab scale experiment, generally we will be doing in the 250 ml flask. From 250 ml flask we can transfer it to 500 ml flask, 1 liter flask or 2 liter flask. For larger scale we will go for pilot plant, and for pilot plant we will go for industrial scale fermenters. We should remember that in each and every step there the fermentation media or the growth media should be free from the contamination. It should free from contamination should be taken care of. Why? Because if you are not following the correct protocol, it may lead to the contamination and all our efforts may be wasted. Next, so after for every enzyme there is a time interval, means the fermentation process may takes for 7 to 10 days, or it may takes place for 20 to 7 to 30 days, or based upon the growth of the microorganism, based upon the excretion of the enzyme. The enzyme is how now after the certain time interval or after certain days, the enzyme has been excreted into the fermentation media. If enzymes, again I am telling, enzymes can be of exocellular enzymes or endocellular enzymes. Now the final step is the recovery of enzymes from the fermented broth. So how do we recover? Generally enzymes can be recovered initially by separation. We need to separate the medium by filtration methods, centrifugation methods. If it is exocellular enzyme or if it is endocellular enzyme. In case of endocellular enzyme, the cells should be disrupted. The disruption process can be takes place with the help of by following a process of centrifugation, where we provide high speeds for disruption of cell, and it tend to release the enzyme. Now after release of enzyme, the sample is been collected and it is purified. As purification should be carried out. Why? Because this cell sample can be of certain impurities. Pure form means the sample should contain only enzyme but not other kind of impurities. Purification can be done with the help of chromatography techniques. So by studying this, every university and in our country and in all throughout the world, many microbiology researchers are working on the different types of enzymes. So I suggest to the students to go for the project work or as a research topic after their BSc, to do in the research in this therapeutic enzymes or other kind of enzymes. Why? Because comparing with the high end prices of drugs for curing the different type of disease, normal people or layman people are not able to purchase these drugs. If we exploit the microorganisms which are available in uncountable number in our normal microflora, we can make use of these enzymes. Enzymes coming from the microorganisms. Now after studying this, at least we should know about what are the most favorable applications or most general applications of therapeutic enzymes. Now why therapeutic enzyme means? Therapeutic means therapy or treatment. Enzyme therapy involves oral enzymes, involves taking the oral enzymes to improve the digestion, immune system and overall health. Now how these enzymes are given to the humans or the patients means in the form of tablets or in the form of syrups or as a injections. Nowadays enzyme therapy has been mostly followed by the many practitioners and many doctors by subsiding the commercial use of tablets and antibiotics. Now, basically therapeutic enzymes are used in the treatment of tumors. So, these therapeutic enzymes are referred as anti-tumor agents. They are used in the treatment of skin ulcers. Here, collagenase enzyme plays a important role in treatment of skin ulcer. Next, for the treatment of several diseases such as leukemia, Pompe's disease, cardiovascular disease, Parkinson's disease, Fabry's disease, digestive disorders and pancreatic disorders. They are also employed in the diagnosis, biochemical investigation and monitoring of many alarming disease. They can be even used as a biomarkers in identification of certain diseases. Generally, traditional methods of ELISA, RIA are used as a biomarkers for identification of diseases. However, here enzymes can be employed for identifying as a biomarker in certain diseases. Apart from this, I will discuss about certain enzymes and their functions. Serratiopeptidase is an enzyme useful in the treatment of pain and inflammation. As the name suggests, peptidase enzyme tend to remove the pain and inflammation by degrading the protein part. And next is a Strepto Kinase. Strepto Kinase in the enzyme is used as a anticoagulant activity. Generally during the blood infections by Staphylococcus aureus, it tend to release the coagulase enzyme. These coagulase enzyme tend to cause the clotting of the blood. So, due to the clotting of blood, there is no free movement of the oxygen and patient may die due to the lack of the oxygen. So, Strepto Kinase enzyme isolated from the microorganisms is having a anticoagulant activity. This anticoagulant activity can it can prevent the self-coagulation of the blood by certain pathogenic bacteria or blood-borne pathogens. Next is the beta-lactamase enzyme. Beta-lactamase enzyme has been given to the patients suffering with the penicillin allergy, some certain penicillin allergy. Generally, certain microorganisms have a resistant ability. Resistant ability means there is no effect of certain antibiotics on them. So, this beta-lactamase enzyme tend to reduce that activity and it enhances the activity of a penicillin antibiotic to kill that particular pathogen. And another is a ribonuclease enzyme. Ribonuclease enzyme, as the name suggests, this enzyme acts especially on the RNA containing viruses. It tend to degrade the nucleic acid of the RNA viruses. Next is enzymes like certain enzymes L-arginase, L-glutamase, L-tyrosinase are employed as a anti-tumor agents. As the name suggests, arginase, glutamase, tyrosinase, these enzymes are acting on those particular amino acids. These amino acids are kind of essential amino acids required for the growth of any particular cell. Due to reduction or due to reducing their ability to synthesize or utilize these amino acids, the tumor cell can be prevented from excessive proliferation. Now, arginase enzyme tend to decreases the availability of arginase enzyme. Glutamase enzyme tend to decrease the availability of glutamine amino acid. Tyrosinase enzyme tend to decrease the availability of tyrosine amino acid. Another group of enzyme is deoxyribonuclease. Deoxyribonuclease is enzyme. It acts as a mucolytic agent in patients suffering from chronic bronchitis. Chronic bronchitis is a disease where there is a excessive accumulation of mucus in the lungs. And generally, patients are subjected for antibiotics for decreasing the level of mucus. However, due to the high cost effective or due to side effects of the antibiotics, Now doctors or practitioners are suggesting for the enzyme therapy. In such cases, deoxyribonuclease acts as a promising agent to reduce the mucus present in the chronic in lungs, patients suffering from the chronic bronchitis. Protease enzyme. Apart from these known enzymes, protease enzyme is a general enzyme. As the name suggests, protease enzyme generally tend to degrade the proteins. For any pathogen to show its action or activity, it uses certain proteins as a antigens in the host. When protease enzyme has been employed as a antibiotic or as a antimicrobial agent, it tend to degrade the important proteins present in the pathogen, especially blood-borne pathogens. Hence, it acts as a blood enhancer. Next is a lysozyme enzyme. As lysozyme enzyme is an enzyme known to each and every one of us. Lysozyme enzyme is generally secreted in the tears, saliva, sweat glands of the human. Lysozyme enzyme, the major role of lysozyme enzyme is a hydrolysis of the cell wall of pathogenic bacteria. Generally, we know that cell wall is a main component or major component of any bacteria. Without the cell wall, the bacteria cannot multiply, or bacteria cannot divide, or bacteria, that pathogenic organism, cannot increase its population. On administration of lysozyme enzyme, we can dissolve or we can break the cell wall of the bacterial pathogens. Next is a glutamase enzyme. Glutamase enzyme is generally used in the treatment of leukemia. So, as we have studied about the types, what are the enzymes, what are the types, what are the production, how these enzymes are produced, and what are the applications of enzymes. It is most interesting and promising subject as a research point of view and as a project work of point of view. Each one of us try to implement these thoughts after studying this topic, that we should go for research in isolating the microorganisms that are producing these kind of enzymes. For this process, for following this research as an interest, it is not so very tough job also. From our general micro soil, we can isolate the enzymes, we can isolate the microorganisms, we can prepare the media, we can isolate the enzymes, we can work on the activity of the enzymes. So, apart from this, we will dealing about the other group of enzymes also. Other group of enzymes such as amylase enzymes. Amylase enzymes are the enzymes which acts upon the substrate, which acts upon the starch as a substrate. Lipase enzyme which acts on the lipid as a substrate. Protease enzymes which acts on the proteins as a substrate. Lactase enzyme which acts on the lactose as a substrate. Apart from for general replication process, even we have polymerase enzymes. Polymerase enzymes which plays important role in the DNA replication. Polymerase enzymes, restriction endonuclease enzymes, their role is in the genetic engineering, where they tend to cut the specific sequences in the DNA fragments. We have endonucleases, we have exonucleases. Endonucleases are the enzyme which produces the internal cuts in the any DNA sequences. Exonucleases are enzymes which produces does a cuts at the exterior point of a DNA sequence. So, now we can wonder that these enzymes are not a chemical preparations, but these enzymes are microbial origin. So, it is very easy for us to isolate these enzymes. So, if we keep on counting, there are innumerable number of enzymes, and all the source of enzymes are nothing but microorganisms. Microorganisms means bacteria, fungi, a certain group of yeast, mold, and even algae. Going apart from this, even enzymes can be commercially also be produced. Commercial in the sense means through the chemical modifications. Chemical modified enzymes are also available in the market, but comparing with those chemically modified enzymes, it is better to use our microorganisms. Since we are studying microbiology, we should make use of these microorganisms, and since microorganisms are present in our surroundings, it is better to exploit this microorganism for benefits of our health, for improving our immune status, and even for the commercial production. Enzyme technology has been exploited, or enzyme technology is a promising future, which is giving a it is serving as $1 billion business throughout the world, and we can also earn certain kind of dollars in our hand through by following these practices. So I want to suggest that it is better or it is a good opportunity to make use of after studying this topic, that okay, enzymes are easily available through microorganisms, and microorganisms are readily available in our surrounding. The only requirement is the growth media. How that growth media should be developed, how optimization of growth media should be done. If we follow those certain small points and certain small things, we can use the enzyme, we can use or we can make business out of the enzymes.
Myself, Murali Mohan Reddy, lecturer in Computer Applications, Government Degree College, Nagari. Before starting our session, we briefly discuss about what are the topics that we have discussed earlier. All of you know what is a variable. Variable is a memory location that is used to store values temporarily for processing, and the variables hold some value that can be used for processing. If you want to store 100 values, what we have to do? For that, we have to declare n different variables, so which makes us difficult for us to process the n variables declared separately. So what we can do, that can be reduced by using the data structures provided in all the programming languages like arrays. So array is defined as in this session we are going to learn about what are the topics, or what is an array, why use an arrays, what are the applications of arrays, what are different types of arrays, and creating arrays. What is an array? Array is defined as a collection of homogeneous elements, or otherwise array is defined as a collection of elements of same data types which are referred with the same name. where the array elements are referred with an index unique index number that is provided for all the array elements. Array is one of the important programming element that are used in programming languages. Why we have to use an array? The contents of array can be stretched. Unlike the variable where we can store only one value, array provides us to store n number of values, as well as array helps us to process the values easily, and it also helps us to write the programs, easy and simple programs. The applications of arrays. The arrays are used to store the list of test scores and sort the elements very easily. These are the some of the applications that are being mentioned here, where there are many other applications that we can that we can use with the help of arrays. Arrays in PHP is a little bit different from the other arrays. Arrays in PHP are the special variables that can store as many values as required, when compared with the other programming language arrays where the array size is limited, whereas in PHP the array size is not limited and we can store any number of values. As well as here in the arrays in PHP, the arrays are considered as an ordered map where the values are maintained in the form of key and values. It is a pair of key and values. Unlike the other programming languages where the array elements are of same types, whereas in PHP the array elements can be of any type, which we call it as a loosely typed language. So the loosely typed languages have that provision where we can assign different value for each variable, unlike the strongly typed languages like C and C++, where we can assign only one value at any time. In PHP, the most of the coding will be for web application development. It is very common for to be working with tabular data, where we can store this tabular data in the pair of nested arrays. By storing the data in pair of arrays, the PHP provides us different many benefits, and it has many built-in functions, library functions, through which we can easily manipulate the lot of amounts of data that can be processed, that we have to process. There are three types of arrays in PHP. They are indexed arrays, associative arrays, multidimensional arrays. Arrays with numeric index are called as indexed arrays. Arrays with named keys are called as associative arrays, and arrays containing more than one array is called as multidimensional arrays. Here each and every arrays has its own benefits as well as some limitations. We will discuss what are the benefits and what are the limitations in the main class. Arrays can be created in two different ways, one using the array function and the other using array operator. Array function is used to create a new array with n number of elements, where we can create an array with more than one element at any time, whereas array operator is used to create an array with just one element, and as well as the array operator is used to add an existing element to the existing array. Indexed arrays, which are also called as arrays with numeric index. Here in this array elements are maintained in the form of key and value, which is generally called as a pairs, where key is the index position, as like we use in the normal programming languages, which starts with 0, and value is whatever the value that we stores in that array locations. And here the indexed arrays also can be used to maintain as many values we want. So here when you consider this array as a list of containers, where each container is having a value and that index starts with 0, as like with the other programming languages, whereas in other programming languages the array elements are stored in a continuous memory locations, and they will be easy for it allows us to easily process. Likewise here the arrays in PHP maintains the data in the form of hash tables instead of the continuous locations. So the index may not be, sometimes we can change the index in any order we want. how to create an indexed array using an array function. So, for that the PHP providing us a function that is called as array function, and with the help of this function we can create an array with n number of elements. This is the code given here for creating an array, where this array consists of elements of different data types, whereas unlike with the other programming languages where the array will be of same type, because PHP is a loosely typed language which gives us the provision to store different type of values. Here the code, here this slide shows how to create an array using an array operator. The array operator is the one where we use the square brackets as like we use in the other programming languages. This array operator allows us to create an element by element, where we can create the array with any number of elements, whereas we can create only, or we can assign only one element at any time. If you see here, we are not giving any index number for assigning the while assigning the values. Though we are not giving any index numbers, the index number, the PHP automatically assigns the index number. So, here the first index will be 0, the second one will be 1, the third one will be 3, and so on. So, here we have an advantage, unlike the other programming languages, we have an advantage that PHP allows us automatically assigns the index number in case of numeric index array. If you consider the above two examples, where in both the cases an array is created with 6 elements, where the first element is having a number and the second and third elements are having integers, and the last element with an index 5, index as 5 and the value as 50. And how to access an array element? As like the other programming languages, the array element can be accessed using the subscript or the index, which generally will be of type array name, and in the square brackets we have to give the index number. With the index we can access any element we want to access. That is why these arrays are also called as subscripted variables. How to add an element to an existing array? We can add an element to the array by just using the array operator. The array operator is nothing but using the square brackets followed by the variable name, and just we have to assign the value whatever you want to assign. How to remove an element from an array? To remove an element from an array, we can use the function unset function, that is a library function which destroys a single element of an array. So, unset and array name followed by the index, the element we want to, or the location which we want to remove must be given in this as an argument in this function. So, this is a sample program that is given here, which allows us to process n number of values easily by just using some kind of looping statements, which is not possible with the normal variables when we use the normal variables to store the values. So, here the student details are being represented as an array, and all the details are being entered here, like roll number, name of the student, as well as the marks that has been obtained by the student. So, in order to process the array elements, we can use a looping statements through which we can add or we can perform all kind of operations like sorting or otherwise finding the maximum, minimum, and other processing of the data. So, here this particular loop where I have used a function to find the number of elements that are present in the array. So, count is the function which gives the number of elements that are present in the array, so that we can easily add all the elements by using a single looping statement instead of using using an operator or n number of variables for adding the elements. So, if you are using an indexed array, if you add an element in the middle of the array, what happens means it will be difficult for the programs which we have written previously. So, what we need to do is we have to change all the programs that are using the structure or the indexes for their processing, which is one of the limitation of the indexed arrays. So, In order to overcome this, the PHP provides us a new type of array that is called as associative arrays. Instead of using the indexed keys, here it uses named keys. Associative arrays use the named keys, where each and every element of an array will be identified with a arbitrary user defined strings, and it also has an advantage that it can store n number of values, that is as many values we require. There is no restriction. So the associative arrays are also created using the array function as well as the array operator. So in order to create an associative array, we have to use the same way of creating the arrays with the array function, but instead of using the index, where the index will be assigned by the PHP automatically, we have to use the key. The key is given by the user. So by giving the keys like this, it will be easy for us to add the array elements or introduce the new values for the keys, so that without changing any other programs that are already been written. So just we need to add whatever the keys that have been added here. So these keys allows us to access the values easily by remembering the key name, whereas in case of indexed arrays, we need to remember all the index numbers. Based on the values, we have to remember the index numbers, and we have to use the appropriate index numbers in the programming. That will be reduced by these user defined keys, where the user can give whatever the key that a user require. And using the array operator also, we can create the associative array, whereas we have to use the array name and the key followed by the value. So this is the way we can create an associative array, and here the key is a literal. We should not give any kind of expressions, but we have to use a literal. To add an element to an associative array, we can use the array operator, where the syntax is given here like this: array name, the key followed by the value which we want to associate into the, or which we want to assign into the associative array. Accessing an array element from the in order to access an element from the associative array, we have to use the same syntax, using the array name followed by the key which as a index. So this is the program where we have defined an associative array and for accessing the values from the associative array. So here this is the keyword that is used for creating an array, as like in case of the numeric array also, indexed arrays also. Here this is the key which is followed by the value, key followed by value, and here this is the operator which assigns the value for the keys, which associates the value for the keys. And in order to access the associative arrays, we have to use the keys, and that should be given as a literals. And here this particular code is used to create an array and to access the elements of an array. And the third type of array we have is multi-dimensional arrays. Unlike the other two types of arrays, these multi-dimensional arrays are also called as an array of arrays. Here each element in the multi-dimensional array itself is an array, or it can be an element. So these multi-dimensional arrays are also created using the array function. Here I have written a program through which we can access the array elements, or we can create the array and we can access the array elements. So here from first we have to use the array function to define the subarrays here. So here this is a subarray 1, subarray 2 and subarray 3 to store 3 student details. So likewise we can store n number of student details. While accessing the array elements, we need to use the array subscript for indicating the rows, as well as array key for accessing the element. These associative arrays helps us to store data that is to be processed when we have large amounts of data that has to be maintained in a form of rows and columns. these associative arrays is generally used for storing the values from the databases. So, these are the 3 different types of arrays we have in PHP. Each and every array has its own advantage and disadvantages. When you take the indexed arrays, we have the data is maintained in the form of in the elements in the form of indexed locations. So, here now we will conclude the class, where in this session we have learnt about the arrays which are capable of storing multiple values. Arrays are flexible when compared with the other programming languages. Arrays in PHP, we can store n number of values, and we have 3 types of arrays: arrays with a numeric index, arrays with associative keys which are also called as descriptive names, and multidimensional arrays where the arrays containing other arrays. Thank you. In the previous session, we have learnt about the arrays. We have different types of arrays, like a numeric array, index, associative array, and multidimensional array. In this session, we learn about what are the different array functions that are available in PHP. So, in order to know the number of elements that are present in an array, the PHP provides us lot of functions like count and size of. Count and size of are the 2 functions which will do, they both will do the same task of counting the number of elements that are present in an array. The count is the function which gives the number of elements that are present in an array, whereas the size is also alias of the count function. The syntax of the count function is like this: count followed by the array name and the mode. Here the mode is the most important one, which gives the number of elements based on the type of array. When we give the default, or if it doesn't give any value for this, the default value will be 0. So, this simply gives the number of values, number of elements that are present in the array, even though it is a associative array, multidimensional array. Whereas, if you want to know the exact number of elements that are present, even it is an multidimensional array, for that we have to use the mode, where by giving the mode as 1, it gives or it counts all the elements that are present in the array, if in case of the associative array. If we give the value as default, that is 0 or otherwise nothing, it gives the number of elements. That means, in case of associative array consisting of 5 elements, it simply gives as 5, where each element can be a sub array. So, in that situation, in order to know the total number of elements that are present in an array, then we have to use the mode as 1, where it recursively counts the number of elements that are present in the array. So, in order to know the count size of the array, we can use either count or size. In the same way, whenever we want to add an element, already we have used the array operator, but the PHP provides us the functions through which we can add the elements at the end of the array. For that we can use the function push. Array push is a function which adds one or more element to the end of an array, existing array. So the syntax of array function is like this: array, where we have to provide the existing name, existing array name as the first argument, and the remaining are the number of elements, where we can give n number of elements to add to the existing array, where the element can be again an array. So this array push function adds the elements to the end of the array. Likewise, this array pop function is the one which removes the last element of an existing array. With the help of this array pop function, we can remove an element, that is the last element that is present in the array. And sometimes we need to add the elements at the beginning of the array. Then what we have to do? So in case of normal programming, it is difficult for adding the elements at the beginning of the array, unlike the adding the elements at the end, because whenever we want to add an element at the beginning, we have to move the elements, or we have to shift the elements from one location to the another location, which is a tedious process. Whereas the PHP provides us some of the library, some of the functions through which we can do the task very easily. Those two functions are unshift and shift. Array unshift is a function which allows us to add one or more element at the beginning of the array. So the syntax of the array unshift function is like this: array unshift existing array elements. So here this shifts the elements towards the right and it adds the elements. Likewise, whenever we want to remove an element from the beginning or from the starting position, then we can use the array shift. This function allows us to remove the first element from the array, whereas from the beginning of the array. Unlike the array pop function, which deletes an element at the end of the function array, the array shift function removes element at the beginning of the array. And whenever we want to arrange the elements, the PHP provides us many functions for processing the entire array at a time. Whenever we want to arrange the elements in an order, the PHP provides us a function that is called as sort. With the help of the sort function, we can arrange the elements in any order we need. By default, the elements will be arranged from lowest to highest. The syntax of the array function is like this: sort is the name of the function, and here we have to use the array which we want to sort, and here we have a sort flags which is an optional parameter which is used whenever you want to sort the elements into a particular type, based on the particular type or in a particular order. We know that array consists of elements of different data types in PHP. Whenever you want to sort the elements, the text elements or string elements will be considered as different with the numeric elements. So in order to sort them, even though of different data types, we have to use this array sort flags. The sort flags generally have the values like sort regular, sort numeric, sort string, sort natural. So here the sort regular option, which sorts the elements in a regular manner, considering both as text as text and string as string. Whenever we want to sort the elements based on their numeric values or based on numerically, then we can use the sort flag sort numeric. Likewise, when we want to sort the elements as a string, even though the values are of numeric type, we want to sort them as a string, then we can use this sort string. Likewise, we can use the other parameter sort natural way that the PHP will sort. So the sort function helps us to arrange the elements from lowest to highest. Likewise, we have another function that is called as array merge, which helps to combine more than one array at a time. So this also helps us to combine the data that is present in more than one array. The syntax of the array merge function is like this: array merge, array name 1 and array 2. Likewise, we can give n number of arrays, where the result of this array merge will be a new array storing all the elements. So for example, if we want to change the elements location or otherwise the elements position. For that, the PHP provides us a function called as a shuffle, which randomizes the elements of a given array so that the elements will be rearranged in a random order. The syntax of this function is shuffle existing array. And sometimes we need to access what are the keys that are present in an array. For that, the PHP provides us a library function that is called as array keys. This is the function which returns all the keys within a given array into as an array. So this helps us to separate what are the keys that are present as well as what are the values that are present can be known with the help of the array values. So these two functions, array keys and array values, helps us to know what are the different keys that are present and what are the different values. So with the help of the keys that are present, that can be used in our programming purpose. So these two functions helps to know the keys and values in case of associative arrays. And sometimes it will be better to transfer the elements as a string. In that situations, all the array elements can be transferred or can be converted into a string. For that, the PHP provides us two functions, one to convert the array elements into a string, the other to convert the string into the array elements. So the PHP is a scripting language where the most of the data processing is done through the web, where we need to transfer large amounts of data, and here when you want to store transfer the values one by one, it will be a difficult process. So in that situations, if you transfer the entire values as a string, it will be a beneficial one. For that, we can use the function that is given that is built in, that is called as implode. Implode is the function which converts the array elements as a string, and the array elements are separated with a delimiter that we will mention in the function itself. So implode delimiter followed by the array name. So here the delimiter can be any symbol that is not a reserved the delimiters that are used for any special purpose. So this function converts the array elements into a string, so that the entire string can be used for transporting from one system to the one application to the another application, where the elements can be again reconverted into the values by using this explode function. So the explode function returns an array of strings, each of which is a substring formed by splitting it on boundaries formed by the string delimiter. So the explode function splits the array into the strings into array elements. So these two functions are very useful for us in case of web developing programming, where we have to transfer the data. And there is another function that is to reverse the array elements. So whenever we want to reverse the elements, we can use the array reverse function, through which we can reverse the entire array, and it returns an array containing the elements that are reversed. And whenever we want to identify what are the elements that are present in an array, for that we can use the function inArray, which helps us to identify whether a value that is present in an array or not. And array unique is the function which is used to remove the duplicate values from an array. So this is also a big problem for identifying the duplicate elements in array in web applications. So most of the time the users enters the data. So in order to remove the duplicate entries, this particular function will be useful, where we can remove the duplicate elements from the arrays, or otherwise the data that is being stored in the form of arrays. So which takes the input array and it finds the duplicate values and it returns an array with unique values. Here also the keys are preserved, and here it uses the sort flags such that based on the type of values, whether the key values must be numeric or strings, that has to be considered. So that will be considered with the help of these flags that has to be given as the second argument, a second parameter for the a unique array function, where this is an optional parameter, and each array has an internal pointer to access the current element. So, as we want to know what is the current position where the internal pointer is, and we want to access the current value, we can use the function current array name. So, here without the index value, without the index or without the keys, we can access the array elements. So, the current function is the one which gives the current value that is present, that is pointed by the internal array. Likewise, we can use what is the next value that is present in an array with the help of the next function as well as next function. This next function advances the internal pointer to the next array element before and gives the next element that is present in the next location. Likewise, whenever we want to move backward, for that we move the internal pointer backward. For that also we have a function that is called as previous, which moves the internal pointer by backward by one step backward, and it gives the previous element that is present in the array. And it also has the function reset whenever we want to move the internal point to the beginning. So, some situations we need to move the pointer to the beginning. For that we can use the function reset. It sets the internal pointer to the beginning of the array irrespective of the position of the internal pointer. Likewise, whenever we want to move the internal pointer to the end of the array, we can use the function end to move the internal pointer to the end of the function. So, these are the references from where we can find the details about the functions in detail. More other library functions that are present, we can visit the php.net website. Thank you. All programming comes down to taking actions with data. So, in PHP most of the data processing will be from the web. So, here the users enter lots of data in a form of through the HTML forms as well as other ways. The data will be stored and it will be processed and it will be sent back to the clients. So, here the data that is stored for processing the data that is maintained, we have different approaches. One of the approach is a procedural programming, in which the focus is on actions, that is how the data is to be processed, and it will be like this: email it, stores it, etc. In case of the other programming approach, that is OOP, the focus is on the data, that is the object oriented programming mainly focuses on the security of the data. So, in order to design an application, we have to think whether it is a procedural programming or an object oriented programming. In PHP, the most of the processing where we need to store the data in a form of in the databases, and we need to process lot of amount of data, as well as we need to maintain the data security. So, in that situations it will be beneficial to use the object oriented programming, which helps us to maintain the data security, where this object oriented programming is which is a customary to group all the variables and functions together as one unit, and what we call as an encapsulation. So, by maintaining the data and functions together, it provides us the data security, so that it also provides us to access the data that is stored within the object by only those functions. And these classes helps us to maintain the modularity of the programming and reduces the repetitive coding process. The most important terms or the terminology that is used in object oriented programming is class and object. So, this already you know, but for our reference again we are discussing about this class. So, a class is described as a template through which we can define or we can describe n number of objects. So, the class is used to define the state and behavior of an object, and this class is used to define the real world object in our programming. So, the class is a logical entity which describes what are the state and the behavior of an object, whereas the object is a functional element which is used to represent the real world entity into the programming, where this object is the one which actually stores the values for the classes. So, class is just the one which describes what should be present there, whereas the object is an instance through which we maintain the values for the objects. So, this object is also called as an instance of object is called as an instance of a class, which is similar to a variable in the programming, whereas in terms of object oriented programming, the variable of particular data type is called as, here in case of class it is called as an object. So, now we are going to discuss about how to create the objects in PHP. There are many built-in objects, but we are going to know what are how to create an user defined objects. So, here in order to create a object, first we need to define what is a class, and then we can create the object. So, these are the different objectives that we are going to learn now: defining the classes, adding properties to a class, adding methods to a class, creating objects from a class, accessing objects methods and properties, setting objects properties, and how to delete an object. So, here I am just concentrating on the syntaxes, how to create and how to access. So, a class is already a class is the collection of both data and functions, which simply called as members. So, data members and member functions. In PHP also, the class is defined with the keyword class followed by the identifier, which is nothing but a class name. So, we have some rules to define the identifiers, which here also in PHP also same. So, we have to use all the we have to follow all the identifiers, and the class names cannot be a reserved word. Class name cannot be a reserved word. Class contains variables and functions which are generally referred as attributes or attributes and methods. So, collectively the class attributes and methods are called its members. The class is defined with the syntax class name class name preceded with the keyword class. In order to add properties to the class, or otherwise in order to add the attributes to the class, we have to use the syntax like this: class name followed by the visibility and the variables, the list of variables. So, here the one important thing we need to remember when compared with the other programming language is that the visibility is compulsory. In case of PHP, the visibility has 3 values: one is public, private and protected. So, public is the visibility mode where the members of a class can be accessible outside the class also, whereas private and protected are the visibility modes in which the members of a class cannot be accessed outside the class, but they can be accessed within the class. Then what is the difference between a private and protected? So, protected is used whenever we want to inherit the class members from one class to the another class, whereas within a class the private and protected both will have the same functionality. So, while defining the members of a class, this is the syntax we can use: class, the name of the class, private, followed by the variable name, scalar variables, and the values. The values here, whatever that we use to assign, they must be of literals. We cannot use the string expressions, but we have to give the literals. And this is the way we can add the members to the data members to the class, and they can be initialized also. And in order to add the methods to a class, these are the methods which access the data that is present in a class. So, the syntax for creating the class methods is like this: class followed by the class name and the visibility, and we have to use the keyword function followed by the function name and the function code. The methods you define within a class are defined just like the functions outside of a class. They can also take the arguments, default values, and the functions also can be of constructors. So, here also for the functions we have to use the same visibility mode. They are private, public, and protected. When we make these functions as private, it will be difficult to access those functions. So, we need to define at least some functions as public, through which we can modify the data that is defined as a private. So, these are the examples through which we created the member function. We have defined the member functions of a class. So, here while defining the member functions, we have to use this keyword. This indicates the members belongs to this particular class. It is must for in the PHP that to access the members within a class also we have to use this keyword. Otherwise, it will show it as an it will be difficult to access the members. And while using this keyword, there is no need to give the $sign before the variable names, which we generally use it to do. So, how to create a object? Objects are created using the new keyword, as like the other object oriented programming languages. So, here we have to use the syntax object name is equal to new class name. So, here the class name is the one we have to give the name of the class which we have created, for which we want to create an object. So, this process of creating an object is also called as instantiation. And as we said, generally this object also can be considered as a variable of a particular type, but in terms of object oriented programming, the variable of a class is called as an object. And how to access the members of a class? So, the members of a object are accessed using the syntax object name followed by the object accessing operator and the method name or the property name, as like in this case method name and property name. Here the method name, whenever we have the values, we can give the values also. So, the methods also takes the arguments, and here we have to remember that in our general normal programming practices, always we put the variable $sign before the variable name in PHP, but in case of accessing the methods or members of a class, we should not give the $sign before the method name or a variable name. So, if you give a $sign before a method name or a data member name, we will get an error because of this particular $sign giving in our practice. And how to set the object's properties? So, the object's properties can be set at the time of declaration, and we can use the object name, property name followed by the value when. So, here I have defined property. So, the public properties, the public members can be accessed in the programming, whereas the private members cannot be accessed. So, they have to be assigned through other member functions. And in order to delete an object, we have to use the function unset, which is used already in the arrays also, where this unset is the function which removes an object from the memory. So, this is a sample program that I have written, through which an object is created, a class is created, defined like with the name of student, class student, and the values are being assigned here. And here there is a function to read the values. So this is the important thing we need to remember, that we have to use this keyword before all the variable members name, even though we are using within the function, within the class member function. So this, with the help of this keyword, the PHP recognize the member name into which it has to assign the values. These member functions can take the arguments as input that can be used here while for initializing the members. So here this will be the default values. If you create an class, create an object of this particular class, these members will be initialized to the with these values by default, even though we have not written any constructor. Whereas if you use this function, this it will add it will assign the values that whatever we have given here. So here this is the function to display the values. If you look here also we have to use this, and here the dot, it is the string followed by a dot, and the dot is the concatenation operator which concatenates the two strings. So in the first case I have created an object and I have just I have not given any values. So this produces the output like this, whatever the default values that we have given here. Whereas in the second case I have called the function and I have given the values, the function, the member function of a class, and I have given the values. In this case the object created is assigned with the values that we have provided, and that has been displayed here. So this is the way we can create the classes. This is the way we can create the classes and we can access the elements. So these are the thank you.
డియర్ స్టూడెంట్స్, ఇంతకుముందు క్లాస్లో మనం లాప్లాస్ ట్రాన్స్ఫార్మ్ని ఏ విధంగా డిఫైన్ చేస్తాము, దాంట్లో ఫస్ట్ షిఫ్టింగ్ థియరం ఏంటి, సెకండ్ షిఫ్టింగ్ థియరం ఏంటి అనేవి మనం నేర్చుకున్నాం. ఇవాళ క్లాస్లో మల్టిప్లికేషన్ బై టీ అండ్ డివిజన్ బై టీ అనేటటువంటి థియరమ్స్, వాటి అప్లికేషన్స్ని మనం స్టడీ చేయడం జరుగుతుంది. ముందుగా మల్టిప్లికేషన్ బై టీ. ఇఫ్ f of t is a function of class A and if L of f of t is equal to f of p, then Laplace transform of t into f of t is equal to minus f dash of p, or Laplace transform of t into f of t is equal to minus d by dp of f of p. So dear students, actually Laplace transform of f of t ante integral 0 to infinity e power minus pt f of t dt. సో ఈ వాల్యూని మనం దేంతో డినోట్ చేస్తున్నాం, f of p తో డినోట్ చేస్తున్నాం. సో అంటే ఇక్కడ f of p is equal to integral 0 to infinity e power minus pt f of t dt. ఇప్పుడు దీన్ని ఇరువైపులా p తో డిఫరెన్షియేట్ చేస్తే దట్ మీన్స్ d by dp of f of p is equal to ఏమవుతుందో చూద్దాం. దట్ ఈస్ d by dp of integral 0 to infinity e power minus pt f of t dt. ఇది. సో ఇక్కడ మనకు ముఖ్యంగా రెండు వేరియబుల్స్ కనిపిస్తున్నాయి. ఒకటి t, రెండోది p. ఇవి రెండు ఒకదానికొకటి ఇండిపెండెంట్ వేరియబుల్స్ కాబట్టి ఈ ఆర్డినరీ డిఫరెన్షియేషన్ కాస్త ఇంటిగ్రేషన్ లోపలికి వెళ్తే ఏమవుతుందంటే పార్షియల్ డిఫరెన్షియేషన్గా చేంజ్ అవుతుంది. కాబట్టి d by dp of f of p is equal to integral 0 to infinity dou by dou p of e power minus pt f of t dt. ఓకే, సో మనం ఇక్కడ e పవర్ మైనస్ ptని pతో పార్షియల్ డిఫరెన్షియేట్ చేస్తున్నాం కాబట్టి ఇక్కడ t అనేది ఏమవుతుందంటే కాన్స్టెంట్ అవుతుంది. కాబట్టి దిస్ ఈజ్ ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ e పవర్ మైనస్ pt ఇంటూ మైనస్ t f ఆఫ్ t dt ఈ విధంగా వస్తుంది. సో దీన్ని నేను ఎట్లా రాస్తున్నానంటే మైనస్ ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ e పవర్ మైనస్ pt t ఇంటూ f ఆఫ్ t ఈ విధంగా అడ్జస్ట్ చేస్తాను. ఇక్కడ లాప్లాస్ ట్రాన్స్ఫార్మ్ డెఫినేషన్ మీరు ఒకసారి కనుక కానీ గమనించినట్లయితే L of f of t is equal to integral 0 to infinity e power minus pt f of t dt. అంటే ఇక్కడ f of t ఉందంటే there is nothing but the Laplace transform of f of t అని. కాబట్టి దీంతో కంపేర్ చేసినప్పుడు ఇక్కడ f of t బదులు ఎంత ఉంది t ఇంటూ f of t. కాబట్టి దీన్ని ఎట్లా రాసుకోవచ్చు అంటే లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ ఆల్రెడీ మైనస్ ఉంటుంది మైనస్ లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ t ఇంటూ f of t అంటే f of p is d by dp of f of p is equal to minus Laplace transform of t into f of t. సో దీన్ని కొంచెం అడ్జస్ట్ చేసి రాసుకుంటే ఎలా ఉంటుందంటే Laplace transform of t into f of t is equal to minus d by dp of f of p. So this is the theorem of multiplication by t. అంటే లాప్లాస్ f of t అనే ఫంక్షన్ని tతో మల్టిప్లై చేస్తున్నప్పుడు వచ్చేటటువంటి ఈ టోటల్ ఫంక్షన్కి మనం లాప్లాస్ ట్రాన్స్ఫార్మ్ కనుక కానీ ఫైండ్ అవుట్ చేస్తే అది దేనికి ఈక్వల్ అవుతుందంటే f of pని pతో డిఫరెన్షియేట్ చేసే వచ్చే వాల్యూకి ఈక్వల్ అవుతుంది. ఓకే, నెక్స్ట్ కమింగ్ టు ది నెక్స్ట్ థియరమ్, దట్ ఈస్ multiplication by t power n. So this is if Laplace transform, that is f of t is a function of class A, and if Laplace transform of f of t is equal to f of p, then the Laplace transform of t power n into f of t is equal to minus 1 whole power n into nth derivative of f of p, or the Laplace transform of t power n into f of t is equal to minus 1 whole power n into d power n by dp power n of f of p. సో ఇప్పుడు ఈ థియరమ్ని ప్రూవ్ చేస్తాం మనం. ఆల్రెడీ ఈ థియరమ్ని ఎలా చెప్పొచ్చు అంటే మ్యాథమెటికల్ ఇండక్షన్ ద్వారా ప్రూవ్ చేయొచ్చు. సో మ్యాథమెటికల్ ఇండక్షన్ ఏం చెప్తుంది, ఫస్ట్ గివెన్ స్టేట్మెంట్ని n is equal to 1కి ట్రూనా కాదా అని టెస్ట్ చేయాలి. టెస్ట్ చేసిన తర్వాత n is equal to 1కి సాటిస్ఫై అయితే నెక్స్ట్ n is equal to kకి థియరమ్ ట్రూ అని చెప్పేసి మనం తీసుకుంటాం. ఆ తర్వాత n is equal to k plus 1కి సేమ్ రిజల్ట్ ట్రూ అవుతుందా కాదా అనేది టెస్ట్ చేయాలి. ఇన్ కేస్ n is equal to k plus 1కి కూడా అది ప్రూవ్ కాగలిగితే దెన్ ది గివెన్ స్టేట్మెంట్ ఈస్ ట్రూ ఫర్ ఆల్ పాజిటివ్ ఇంటెగ్రల్ వాల్యూస్ ఆఫ్ n అని చెప్తాం. సో ఆ కండిషన్ ప్రకారం ఇక్కడ నేనేం చేస్తున్నానంటే సో Laplace transform of t power n into f of t is equal to నేనేమని ప్రూవ్ చేయాలి మైనస్ 1 హోల్ పవర్ n ఇంటూ దట్ ఈస్ d పవర్ n బై dp పవర్ n ఆఫ్ f ఆఫ్ p, దీన్ని ప్రూవ్ చేయాలి. సో ఇప్పుడు ఈ థియరమ్ని నేను ఎట్లా ప్రూవ్ చేస్తున్నానంటే మ్యాథమెటికల్ ఇండక్షన్ ద్వారా ప్రూవ్ చేస్తాను. సో మ్యాథమెటికల్ ఇండక్షన్ ద్వారా ప్రూవ్ చేయాలంటే ఫస్ట్ n is equal to 1 తీసుకుంటాను. సో n is equal to 1 తీసుకుంటే ఈ స్టేట్మెంట్ ఏ విధంగా మారుతుంది, లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ t ఇంటూ f ఆఫ్ t is equal to minus 1 whole power 1 into d power 1 by dp power 1 of f of p. ఇది సో ఆల్రెడీ ఇక్కడ నేను ఇంతకుముందే ప్రూవ్ చేస్తున్నానండి మల్టిప్లికేషన్ బై t అనేటటువంటి థియరంలో దట్ ఈస్ ఈ రిజల్ట్ ఆల్రెడీ ప్రూవ్ అయిపోయింది. అంటే దట్ మీన్స్ లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ t ఇంటూ ఎఫ్ ఆఫ్ t ఇస్ ఈక్వల్ టు మైనస్ d బై dp ఆఫ్ ఎఫ్ ఆఫ్ p. ఇది ఆల్రెడీ ప్రూవ్ అయిపోయింది. దట్ మీన్స్ ది గివెన్ స్టేట్మెంట్ ఈజ్ ట్రూ ఫార్ n ఇస్ ఈక్వల్ టు 1. నెక్స్ట్ ఇప్పుడు నేను ఏమని తీసుకోవాలి, ది గివెన్ స్టేట్మెంట్ ఈజ్ ట్రూ ఫార్ n ఇస్ ఈక్వల్ టు k. అంటే అప్పుడు ఏమవుతుంది, లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ t పవర్ k ఇంటూ ఎఫ్ ఆఫ్ t ఇస్ ఈక్వల్ టు మైనస్ 1 హోల్ పవర్ k ఇంటూ d పవర్ k బై dp పవర్ k ఆఫ్ ఎఫ్ ఆఫ్ p. ఇది k ఇస్ ఈక్వల్ టు n ఇస్ ఈక్వల్ టు k కి ట్రూ అని తీసుకుంటున్నాను. నెక్స్ట్ ఇప్పుడు నేను ఏమని ప్రూవ్ చేయాలంటే ది సేమ్ స్టేట్మెంట్ ఈజ్ ట్రూ ఫార్ n ఇస్ ఈక్వల్ టు k ప్లస్ 1 ఆల్సో. అంటే ఫస్ట్ ఇక్కడ చూడండి, సేమ్ ఈ కండిషన్ ఇక్కడ రాస్తున్నాం. దట్ ఈస్ మైనస్ 1 హోల్ పవర్ k d పవర్ k బై dp పవర్ k ఆఫ్ ఎఫ్ ఆఫ్ p. అంటే అర్థం ఏంటి, దట్ ఈస్ లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ t పవర్ k ఇంటూ ఎఫ్ ఆఫ్ t. దీన్ని నేను ఎట్లా రాసుకుంటాను, ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ e పవర్ మైనస్ pt ఇంటూ t పవర్ k ఇంటూ ఎఫ్ ఆఫ్ t dt. ఓకే, సో దిస్ ఇస్ ద డెఫినేషన్ ఆఫ్ లాప్లాస్ ట్రాన్స్ఫార్మ్. నెక్స్ట్ ఇప్పుడు అగైన్ డిఫరెన్షియేట్ విత్ రెస్పెక్ట్ టు p ఆన్ బోత్ సైడ్స్. అంటే ఇప్పుడు ఏమవుతుంది, మైనస్ 1 హోల్ పవర్ k ఇంటూ d బై dp ఆఫ్ d పవర్ k బై dp పవర్ k ఆఫ్ ఎఫ్ ఆఫ్ p is equal to d by dp of integral 0 to infinity e power minus pt t power k into f of t dt. ఇది ఓకే. సో ఇక్కడ నేను దట్ ఈస్ pతో డిఫరెన్షియేట్ చేస్తున్నాను. ఆల్రెడీ k టైమ్స్ డిఫరెన్షియేట్ చేయబడింది కాబట్టి k టైమ్స్ అగైన్ ఇంకొకసారి అంటే టోటల్ ఇది ఎన్నిసార్లు అవుతుంది, దట్ ఈస్ మైనస్ 1 హోల్ పవర్ k ఇంటూ d పవర్ k ప్లస్ 1 బై dp పవర్ k ప్లస్ 1 ఆఫ్ ఎఫ్ ఆఫ్ p ఇస్ ఈక్వల్ టు సేమ్ ఇందాక చెప్పినటువంటి ఆర్గ్యుమెంటే ఇక్కడ మళ్ళీ చెప్తున్నాను. దిస్ ఈజ్ ఆర్డినరీ డిఫరెన్షియేషన్. ఇది ఇంటిగ్రేషన్ లోపలికి వెళితే ఆల్రెడీ ఇక్కడ t అనేది ఒక వేరియబుల్ ఉంది, p అనేది ఇంకొక వేరియబుల్ ఉంది. కాబట్టి ఇవి రెండు ఏమైపోతుందంటే ఇండిపెండెంట్ వేరియబుల్స్ అవుతాయి. కాబట్టి ఈ ఆర్డినరీ డిఫరెన్షియేషన్ లోపలికి వెళితే ఏమవుతుందంటే పార్షియల్ డిఫరెన్షియేషన్గా చేంజ్ అవుతుంది. అంటే ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ డో బై డో p ఆఫ్ e పవర్ మైనస్ pt ఇంటూ t పవర్ k ఎఫ్ ఆఫ్ t dt. ఓకే, సో ఇక్కడ మళ్ళీ e పవర్ మైనస్ ptని డిఫరెన్షియేట్ చేసినప్పుడు ఈ వాల్యూ ఏమవుతుందంటే దట్ ఈస్ ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ e పవర్ మైనస్ pt ఇంటూ మైనస్ t ఇంటూ t పవర్ k ఎఫ్ ఆఫ్ t dtగా మారుతుంది. సో ఆల్రెడీ ఇక్కడ t ఉంది, ఇంకొక t పవర్ k ఉంది, ఇంకొక t వచ్చింది. కాబట్టి t ఇంటూ t పవర్ k అనేది ఏమవుతుంది, t పవర్ k ప్లస్ 1. ఈ మైనస్ బయట రాసేస్తే మైనస్ ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ e పవర్ మైనస్ pt t పవర్ k ప్లస్ 1 ఇంటూ ఎఫ్ ఆఫ్ t dt. సో దిస్ వాల్యూ ఈజ్ నథింగ్ బట్ మైనస్ ఇదేమైపోతుంది బై ది డెఫినేషన్ ఆఫ్ లాప్లాస్ ట్రాన్స్ఫార్మ్. దీన్ని ఎట్లా రాసుకోవచ్చు అంటే లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ t పవర్ k ప్లస్ 1 ఇంటూ ఎఫ్ ఆఫ్ t dt, ఎఫ్ ఆఫ్ t. ఓకే, సో ఆల్రెడీ నాకు దీనికి లెఫ్ట్ హ్యాండ్ సైడ్ ఏముంది, మైనస్ 1 హోల్ పవర్ k ఇంటూ d పవర్ k ప్లస్ 1 బై dp పవర్ k ప్లస్ 1 ఆఫ్ ఎఫ్ ఆఫ్ p is equal to minus Laplace transform of t power k plus 1 into f of t ఉంది. సో బోత్ సైడ్స్ నేను మైనస్తో మల్టిప్లై చేస్తే మైనస్ 1 హోల్ పవర్ k ప్లస్ 1 d పవర్ k ప్లస్ 1 బై dp పవర్ k ప్లస్ 1 దట్ ఈస్ k ప్లస్ 1 ద డెరివేటివ్ ఆఫ్ f of p is equal to Laplace transform of t power k plus 1 into f of t. So that means the given statement is true for n is equal to k plus 1. Okay, so n is equal to 1 కి ఆల్రెడీ ప్రూవ్ అయింది, n is equal to k కి నేను ట్రూ అని తీసుకున్నాను. నెక్స్ట్ n is equal to k plus 1 కి కూడా నేను థియరమ్ని ప్రూవ్ చేయగలిగాను. కాబట్టి ది గివెన్ స్టేట్మెంట్ ఈస్ ట్రూ ఫార్ ఆల్ పాజిటివ్ ఇంటెగ్రల్ వాల్యూస్ ఆఫ్ n. కాబట్టి ది స్టేట్మెంట్ ఈస్ ట్రూ. నెక్స్ట్ ఒక చిన్న ప్రాబ్లం చూద్దాం. సో Laplace transform of t into cos at, దీని వాల్యూ నేను ఎంతని ప్రూవ్ చేయాలంటే p స్క్వేర్ మైనస్ a స్క్వేర్ బై p స్క్వేర్ ప్లస్ a స్క్వేర్ హోల్ స్క్వేర్ అని చెప్పాలి. సో ఇక్కడ దీన్ని నేను మల్టిప్లికేషన్ బై t అనే థియరంను అప్లై చేస్తే ఇక్కడ t ఇంటూ f ఆఫ్ t అనే ఫార్మాట్లో ఉంది కాబట్టి f ఆఫ్ tని ఎంత అనుకుంటానంటే cos at అని తీసుకుంటాను. సో బోత్ సైడ్స్ లాప్లాస్ ట్రాన్స్ఫార్మ్ అప్లై చేస్తే లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ f ఆఫ్ t is equal to Laplace transform of cos at, that value is p by p square plus a square. సో మనకు ఆల్రెడీ మల్టిప్లికేషన్ బై t అనేటటువంటి థియరం తెలుసు. దాంట్లో ఏం చెప్తుంది t ఇంటూ f ఆఫ్ t అని ఉంటే దాన్ని మైనస్ d బై dp ఆఫ్ f ఆఫ్ p గా రాసుకుంటాం. అంటే ఇక్కడ వచ్చిన వాల్యూని నేను దేంతో డినోట్ చేస్తున్నానంటే f ఆఫ్ pతో. కాబట్టి ఇప్పుడు ఏమవుతుంది లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ t ఇంటూ కాస్ at అంటే మైనస్ d బై dp ఆఫ్ దట్ ఈస్ f ఆఫ్ p వాల్యూ ఎంత p బై p స్క్వేర్ ప్లస్ a స్క్వేర్. సో దిస్ ఈజ్ మైనస్ దీన్ని u బై v ఫార్ములా ప్రకారం డిఫరెన్షియేట్ చేస్తే p స్క్వేర్ ప్లస్ a స్క్వేర్ హోల్ స్క్వేర్. నెక్స్ట్ ఇక్కడ p స్క్వేర్ ప్లస్ a స్క్వేర్ రాస్తున్నాను. pని pతో డిఫరెన్షియేట్ చేస్తే 1 అవుతుంది మైనస్ p ఇంటూ ఇప్పుడు p స్క్వేర్ ప్లస్ a స్క్వేర్ని డిఫరెన్షియేట్ చేస్తే 2p ప్లస్ 0 అవుతుంది. సో దట్ మీన్స్ మైనస్ ఆఫ్ p స్క్వేర్ ప్లస్ a స్క్వేర్ మైనస్ 2p స్క్వేర్ బై p స్క్వేర్ ప్లస్ a స్క్వేర్ హోల్ స్క్వేర్. సో దట్ మీన్స్ ఆఫ్టర్ సింప్లిఫికేషన్ I get p square minus a square by p square plus a square whole square. Okay. So the Laplace transform of t into cos at is equal to p square minus a square by p square plus a square whole square. Okay. Next, division by t theorem. So ఇక్కడ థియరం స్టేట్మెంట్ ఏంటంటే ఇఫ్ ద లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ f ఆఫ్ t is equal to f of p, then the Laplace transform of f of t by t is equal to integral p to infinity f of x dx. ఆల్రెడీ ఇప్పుడు మనం నేర్చుకుంది ఏంటంటే మల్టిప్లికేషన్ బై t, ఇప్పుడు నేర్చుకోబోతున్నది ఏంటంటే డివిజన్ బై t. సో అది ఎలా ఉంటుందో చూద్దాం. యాక్చువల్గా ద లాప్లాస్ ట్రాన్స్ఫార్మ్ ఆఫ్ f of t is equal to integral 0 to infinity e power minus pt f of t dt. ఈ వాల్యూని దేంతో డినోట్ చేస్తాం జనరల్గా f of pతో డినోట్ చేస్తాం. అంటే దట్ మీన్స్ f of p is equal to integral 0 to infinity e power minus pt f of t dt. సో ఇప్పుడు దీన్ని బోత్ సైడ్స్ ఇంటిగ్రేటింగ్ విత్ రెస్పెక్ట్ టు p అండ్ టేకింగ్ ద లిమిట్స్ యాజ్ p టు ఇన్ఫినిటీ. సో దట్ మీన్స్ ఇదేమవుతుంది ఇంటగ్రల్ p టు ఇన్ఫినిటీ f ఆఫ్ p dp. అంటే దట్ మీన్స్ f ఆఫ్ pని నేను pతో ఇంటిగ్రేట్ చేస్తున్నాను. నెక్స్ట్ p యొక్క లిమిట్స్ ఏంటంటే p టు ఇన్ఫినిటీ అని కన్సిడర్ చేస్తున్నాను. అప్పుడు ఏమవుతుంది ఇంటగ్రల్ p టు ఇన్ఫినిటీ అండ్ అగైన్ ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ e పవర్ మైనస్ pt f ఆఫ్ t dt అండ్ dp. సో ఇక్కడ ఇది డబుల్ ఇంటిగ్రేషన్. నెక్స్ట్ ఇంటిగ్రేషన్ బై పార్ట్ దట్ ఈస్ మల్టిపుల్ ఇంటగ్రల్ని స్ప్లిట్ చేసి రాసుకుంటే ఇది ఎట్లా రాసుకోవచ్చు అంటే ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ ఈ అండ్ ఇంటగ్రల్ p టు ఇన్ఫినిటీ e పవర్ మైనస్ pt dp అండ్ f ఆఫ్ t dt. సో ఇక్కడ p టు ఇన్ఫినిటీ అనేది ఈ pకి సంబంధించింది అండ్ మోర్ ఓవర్ p అనేది ఇక్కడ ఉంది కాబట్టి దీన్ని మాత్రం ఇంటిగ్రేషన్ చేస్తే సరిపోతుంది. సో ఇప్పుడు దీన్ని ఇంటిగ్రేషన్ చేస్తే ఇంటగ్రల్ 0 టు ఇన్ఫినిటీ ఈ వాల్యూ ఏమైపోతుంది ఇంటగ్రల్ మైనస్ pt బై మైనస్ t. బికాస్ ఇక్కడ మనం pతో ఇంటిగ్రేషన్ చేస్తున్నాం కాబట్టి t అనేది కాన్స్టెంట్ అవుతుంది, అందువల్ల బై మైనస్ t. బట్ ఇన్ ద లిమిట్స్ p టు ఇన్ఫినిటీ f ఆఫ్ t dt. సో ఇక్కడ నేను అప్పర్ లిమిట్ ఇన్ఫినిటీ తీసుకుంటే p బదులు ఇన్ఫినిటీ తీసుకుంటే e పవర్ మైనస్ ఇన్ఫినిటీ అవుతుంది. సో e పవర్ మైనస్ ఇన్ఫినిటీ అంటే దట్ ఈస్ ఇంటగ్రల్ 0 to ఇన్ఫినిటీ e పవర్ మైనస్ ఇన్ఫినిటీ